on introducing real applications of PyData. The first one is Chloe-Agathe Azencott. She's a researcher. She's been doing Python, I think, for 10 years. And she's using PyData actually to help improving our health, if I understand correctly. So she'll speak for 15 minutes. That's a brief talk.
So thanks for the introduction. I'm part of a research group, so I'm actually in academia, so it's a bit weird to be in a real-world session. I'm not used to that. So I'm part of a research group that's called the Center for Computational Biology, and that's a joint research unit between an engineering school in ParisTech and a cancer research center, Institut Curie. And so we work on machine learning for computational biology, and in particular with a focus on cancer applications. So thankfully for me, Gaël has introduced machine learning this morning, so I don't need to do that anymore. And my goal here today is to show you some examples of the type of problems we work on and how I find Python very amenable to solving those problems. So I'm going to narrow down the focus a little bit and have my buzzword of the day is not big data, it's precision medicine. So you might have heard of it under the name personalized medicine, and it's this idea that of moving from a one-size-fits-all approach to medicine where you give every patient that has the same disease the same treatment to an approach that's more tailored to the specific characteristics of each of those patients. And so I'm particularly interested in tailoring it to the genetic characteristics of the patients. And so the idea is to, in the end, give treatment to a person only if you know it's going to work for them, you know it's not going to have dire side effects, and those kind of things. So it's interesting for this community here because it's essentially data-driven biology, in that the main part of it is to identify in data, genomic data from patients, identify the features, descriptors, genomic regions, as you want, that are, shared between individuals that share a same response to a treatment, for instance, and that differentiates them from the people who do not share that response. And then from then on, you also want to build predictor or classifiers that are called in the clinic companion tests, and that will help the clinicians decide which treatment to give to whom. So where do you get data for this kind of problems? Of course, through collaborations, but the example I want to talk about today is through challenges organized by something that's called the DREAM Challenges, so the URL is on my slides. So it's a bit like the Kaggle challenges in machine learning, if you're familiar with those, except that it's non-for-profit and it's focused on questions that are really at the edge of problems that we don't know how to solve and that are very important to human health. So they focus indeed on, they emphasize that we have to deal with large amounts of complex data, and that it's critical to develop new methods to analyze them because we don't know how to do that. So I took part in 2 of those challenges myself, and I want to elaborate a bit on that experience. So the first challenge I took part in was the summer of 2013, so it's a short period. You have 6 weeks, 8 weeks to work on one of those. They give you longer, but by the time you decide you're going to enter the challenge and you take into account everybody's vacations, you have 6 weeks. So this one was on toxicogenomics, genetics actually, sorry, same thing. And the idea is the following: you have a number of cells that have been exposed to a number of chemicals, and each time you measure how toxic these chemicals was for each of the cells. And the goal is to predict whether a chemical you don't know is going to be toxic and for which of those cells, and conversely, for a new cell, is a chemical you know going to be toxic for it or not. So that has lots of applications, in particular in cancer research, to try to target tumors and not the healthy individual. The other challenge I took part in was last year, It was focused on rheumatoid arthritis, which is a very painful disease that has very unsuccessful rates of treatment. So many patients don't respond at all to the treatment they receive. So here you had several thousands of individuals who had received one treatment among 6, and you knew whether they had responded well to that treatment or not. And the question was, can you predict from their genomes whether or not they're going to respond well can you make those kind of predictions for a new patient? So in both those challenges, you have very large amounts of features, so genomic descriptors. So I'm talking millions of features. You have very small number of samples, so of the order of thousands. So comparatively, it's very small. So that creates all sorts of challenges, and that's part of what makes those problems really hard to solve. So I was in both challenges, part of a team that ranked second on some aspect of the challenge. But I can tell you that no team achieved any result that were anywhere near what I would call good. So it's still not something that is usable today in the real world. So to move on to how do we use Python in that context? Well, here are typically the tasks you want to address when you're looking at those type of problems. There's a lot of pre-processing of the data. So Ian actually talked about cleaning data, so I won't go over that again. And then, so as I said, we have a lot of features, so you want to work with fewer features than that. So you'll have to use feature selection algorithms. And then you have to make a prediction. So you want to use classification if you have to make a binary prediction, or regression if you want to make predict a continuous variable. So you have to use classification or regression algorithms. And throughout the whole process, you want to be visualizing what it is that you're doing. And so to go back to those algorithms, machine learning algorithms, whether for feature selection or classification and regression that you want to use, what's going to happen is that you're going to start on the challenge and you're going to be super excited and have plenty of fancy ideas of really smart things you can do. But actually you want to confront those ideas to the baseline, well-proven algorithms that that are known to work to some extent on this type of data. Well, not on this type of data, but in any type of data. And so it will come as no surprise that we've used Python, we've used NumPy, we've used scikit-learn, and we've used matplotlib. And I'll try to give you a few more details about that. So the data pre-processing part, I won't talk about normalization or cleaning the data because it takes too long. I just wanted to mention we've been working with HDF5 files, so through the module H5Py. And one of the reasons for that is that I was able to manipulate that easily through NumPy arrays, and my colleagues that were on the same team and were working with other distributions and other languages were also able to use those. Because a challenge that you start and that's going to last 6 weeks is not something, so it's not the right time to try to convince your lab mates that they should start using R and they should stop using R and MATLAB and start using Python. So you want to give them data they can work with. Regarding feature selection, you'll essentially have two types of approaches. On the one hand, you want to make use of prior biological knowledge that is related to your problem. That's going to require a lot of text processing, going into databases, those kind of things that are related to what Ian talked about before. And you'll also have statistical approaches. You want to use features that are correlated with your outcomes, those type of things. And for that, you can start from scikit-learn and then implement on top of that approaches that you deem fit for your data. The classification and regression approaches, obviously scikit-learn again, with a nice advantage that you can customize your distances or similarity measures or kernels to fit the specificities of your problem. What I really like about scikit-learn is that it automates all those nasty cross-validation and metric computation things which are where I was introducing bugs during the 4 first years of my PhDs. And so we don't have to deal with that anymore. That's very nice. We've also used something that's called Gaussian processes, and for that, so that's not part of scikit-learn, it's not, yet a classical machine learning approach. So we've used GPy, which is an implementation that comes from a lab in Sheffield, and we've also used some custom code on top of that for specific things that were not implemented there. And the last thing I wanted to mention, because I've been running through that talk, was visualization. So when you work with data, I guess I don't have to tell you that, but it's good to get a feel of what it is that you're doing, other than looking at numbers on the screen. So we're very used to plotting results and performance measures to decide which algorithm is better than the other and those kind of things. It's also very important, especially when it's new data of a type that you don't know, you've never worked with those type of things before, to plot things throughout the entire pipeline that you're putting in place. What is your normalization doing? Are all your features the same? Is your similarity matrix different from an identity matrix? Those kind of things. Okay, so I hope I gave you a very brief overview of the type of problems we have to work with in bioinformatics and why there's a lot of space for Python to be used in there. If you're very, very interested in that, we have openings in the group for interns and PhD students. So you can come talk to me. You can come talk to me if you just want to talk about science too. I'm here the whole day, I'm on Twitter, you'll find me. And to finish, I wanted to thank my Python partners in crime over those 2 challenges, so Felipe, Dominic, and Victor, and you for your attention.
Thank you.